Bananas are the 8th most important food crop worldwide and the 4th most important among the world's least developed countries. Bananas are a cash crop, relied on by many as a source of income and many others as a source of food. This chart here shows the leading producers of bananas worldwide, India leading, and shows the thousands of metric tons produced each year. This all shows how important bananas are for the global economy. Panama disease is a deadly incurable pathogen that affects banana plants worldwide, and it spreads very fast. Seen in the map here are all the countries infected by this disease, Europe excluded due to them not having any banana plantations pretty much, but you can see just how much it spread. Because this disease spreads very fast and is incurable, many plantations can be wiped out within months by this disease. Also, hundreds of millions and even billions of dollars are lost in damages each year due to this disease. Seen in this timeline, you can see the years and which countries it spread to during that year. Most notably, this past in 2021, Panama disease spread to South America and has started infecting plantations there. So here's what it looks like when the disease actually infects plants. The leaves yellow and the stem insides become blackened. This is because the fungus is actually stopping the plant from getting the nutrients and water it needs to live. So in an attempt to prevent this disease, scientists created resistant subcultivars of bananas. Seen here, there's many species. These resistant subcultivars were, created to not be able to be affected or be immune to Panama disease. You can see here, Cavendish is the main species of bananas today. They were created as a resistant subcultivar. However, these species are losing their ability to resist diseases because of the genetic mutations that happen in the pathogen that causes Panama disease. The deep learning model itself was trained with the technique of transfer learning. In transfer learning, the base layer is a pre-trained model, in this case the residual net model with 50 layers, or ResNet 50 for short. This specific model is actually used for a lot of plant disease identification tasks. So this is set as the base layer, and it's set to non-trainable to preserve learned information. And then new layers are built on top of this, and these new layers are the layers that are actually being trained during training. This code here is used to import all the packages needed for this program to run. This code here is used to process the dataset and split it into training and validation sets. This code here uses data augmentation to expand this dataset. This code here sets the base layer to ResNet 50, and it makes it not trainable because this is the transfer learning approach that we are using. This code here sets the layers of the actual model itself. You can see that the activation is set to sigmoid, which looks like this, and this activation is used in binary classification tasks like this one. Here's a diagram of the layers of the model. You can see the input, the sequential, ResNet 50, global pooling, dropout, and dense layers. This code here was used to train the actual model itself. You can see the outputs after every epoch, showing the loss and the accuracy values for the training and validation sets. This code here was used to plot these actual metrics. This code here is used to evaluate the model's accuracy on the test set. You can see that's processed right there with that middle code, and the bottom prints the evaluation results. This code here is used to fine-tune the model by training the model, the entire model now, at a lower learning rate. You can see the trainable of the base is true because we're training the entire model this time, not just the new layers. You can see on this accuracy values graph here that the accuracy increases as training progresses and generally converges around 99 to 100% mark. On this losses values graph here, you can see that the losses generally decrease as training goes on and gets very close to zero at the end there. In this confusion matrix, you can see that 245 of the disease pictures were correctly identified and 247 of the healthy pictures were correctly identified. This means 492 out of 500 pictures were correctly identified. In this classification report, you can see the high precision for healthy and Panama disease images with a high F1 score. This means that the model performed with high accuracy throughout evaluation. Here's a video demonstrating how the entire Panama disease detection system actually operates when it's scanning a banana plant. So this project demonstrated that the system was able to identify whether a plant was diseased 98% of the time with very few errors. In addition, considering the high F1 score, which is a metric used in these binary classification tasks, it can also be concluded that the model met the engineering goal that was set. Lastly, it can be concluded that there is a strong natural correlation in the training data due to the low amount of training time needed for the model to achieve this high accuracy. This just means that the images in the training set had a clear correlation to their class, most likely because the Panama disease leaves are very yellow compared to the healthy leaves, which are very green.